That's right. Which is always strange. Okay, it should work. Let me see. Yeah, the bitrate looks fine. And I think we're live. Is the sound okay for whoever is watching us? Let's see, Leo is here.
Oh, excellent.
All right. All right, then. Well, let's get started. Thank you, everyone, for joining us again, like every month. And some of you live, and some of you will watch this later on YouTube. This is a monthly event, monthly or so, to think about what intelligence is all about and what parts of science can help us answer our questions about all those mechanisms and algorithms to understand what intelligence is, not only in our brains, but also in general. So this is organized by Cross Labs, which is a research institute in Kyoto and Tokyo. We really want to create a new kind of university, and these events are part of that. So you can check out our research on, I will post the link, crosslabs.org. There you go. And what we do is really distill the knowledge from different disciplines of the mind, brain science, mathematics, information dynamics, AI, neural nets, cognition, artificial life also, and bring new technology and changes in society to an open discussion. So this Cross Labs is affiliated with Cross Compass, an AI company, and these events are co-organized by the founder of our labs, Antoine Pasquale, Katsunobu Suzuki, and myself. Yeah, I want to thank, as always, our volunteers and Stephen Way and others for contributing to the backstage side of the preparation. And tonight, or today, I have the pleasure to introduce Nathanael Aubert-Kato, who is currently an assistant professor at the Department of Information Sciences from Ochanomizu University. And Nat and I go way back. We actually took our PhD together with the same grant at the University of Tokyo. We also had our paths cross again at ELSI in research about the origins of life. Maybe I'll say a little bit about that, since what you're going to present tonight, I think, is going to be related to your work back then.
Right.
And yeah, the presentation tonight is a bit related to the presentation last month because it relates to the serendipity really about research and intelligence, and especially Nathanael is specialized in DNA computing, molecular programming systems, but also modeling them, simulating them, and developing new smart materials also, which can be linked to a lot of different applications at the end of the day. And the way you get to those applications is not only by researching very narrowly how to develop them, but also research a bit more broadly and with a, I guess, more open-ended kind of method that includes some accidents. So yeah, Nat, whenever you're ready, please feel free to share your slides. And
so
I'm going to
share my screen. So I hope you can see the screen okay. So thank you for the introduction. So as mentioned, I am Nathanael Aubert-Kato. I am currently working in Ochanomizu University, but the work I'm going to talk about was done mostly between the University of Tokyo and ELSI, as Olaf mentioned. And yeah, so it's titled Unexpected Interactions Between Molecular Agents, in the sense that Well, I'm going to present things that happen when things do not go as planned. So usually when we do research, we have some kind of hypothesis, some kind of target in mind, and life, and not quite life, finds a way, right, to do something else. So first, I guess I'd like to talk a bit about molecular programming, in the sense that it's probably not well known by most of the audience. So molecular programming is what happens when you try to interact with systems at the molecular level. So the idea is that you take a bunch of molecules, their concentration can encode data, chemical reactions between those molecules encode operations in terms of normal programming, and the whole network of chemical reactions becomes a program. What is interesting about working at the molecular level is that you have a lot of molecules, like even when we're talking about concentration, that means you have millions of entities, right? But obviously, this is a very different paradigm than standard computing, right? So programming this kind of system can be challenging. And you have to really think carefully about the way everything is interacting. You usually try to remove all the unwanted interactions, because if you don't do that, strange things happen, right? And well, usually presentations that I make are about the times things go right, but sometimes it's interesting to look at what happens when things go wrong. So that's what I'm going to talk about today. So using molecular programming, well, I mentioned molecular agents. So what's a molecular agent? There are many different ways to implement them, but the way I usually do it is by having this kind of molecular robots. We have small beads that are small enough to move through Brownian motion in the environment. So it's kind of random movement. They have a controller, which is a chemical reaction network, as I mentioned. So it's implemented through molecules that are grafted on the surface of those beads, plus signaling and sensing and communication, which is done through molecules diffusing in the environment. So those molecules are produced by those on the beads. So that's signal emission. And at the same time, nearby beads can capture those molecules and react to them. So that's sensing. And finally, we have actuation. So on top of movement, you can also decide to stop by emitting a specific signal that will bind you to your neighbors. And the one example of application is to sense signal from the environment, either those produced by your neighbors or those present in the environment, and decide to assemble into, for instance, a specific shape. But there's all the process to getting to this final implementation, well, this current implementation of molecular robots. And I'm going to talk about, in this talk, mostly the controller part and the sensing part. So the motivation for this specific implementation is to have, well, large-scale swarms, right? So when you have large scale of, a lot of individuals interacting together in a swarm, they can make some interesting structure, especially dynamic structures. And some results suggest that the more individuals you have, the more agent you have, the more complex those behaviors can be. So by going at the molecular level, we can scale to those theoretical results, possibly. And for that, we are using specifically DNA computing. So the idea of molecular programming is kind of general. There is no reason to use specifically DNA, but DNA computing as a candidate for molecular programming has been one of the most popular choice, in the sense that DNA is quite easy to use as a program. So interactions between DNA molecules can be easily set based on the sequence. So complementary sequence will interact together, and unrelated sequence will usually not interact. So it's kind of easy to specify what should interact with what. So the idea is that even in DNA computing, you would have many different programming languages, which are, so, this basic idea of interaction between DNA molecules stays the same. Then you can choose how they're going to react. So a popular approach, for instance, relies on DNA only and use strand displacement. So you have one molecule that will take the place of another, and that cascade of reaction creates the program. In my case, I tend to focus on something called the DNA Toolbox, which uses enzymes to perform the operations. So the DNA Toolbox works like this. So you have your DNA molecule that is part of a simple chemical reaction network, in which a signal DNA strand will interact with a template DNA strand. The green part is complementary with this green part, so they attach to each other. A first enzyme called polymerase will extend that green strand to make a copy of the orange strand. Then a second enzyme called nucleic will cut right in the middle of the top strand. And since this structure is not stable, eventually both the green strand and the orange strand will fall off. So using this template, you can go from a green signal to having both green and orange. To keep things dynamic, we have a third enzyme called exonuclease, which will consume both signals. However, the template is chemically protected against degradation and remains stable. In that sense, based on the previous definition of molecular programming, those signal strands are the data, and the templates are part of the program. And because we have polymerase, exonuclease, and nuclease, we have PEN DNA Toolbox. Right. So this small chemical reaction network actually forms one operation in the program. So at the program level, we just represent it as one arrow going from green to orange. And in the same way, we can have an arbitrary number of operations. In that sense, we can define a program as having an input. So that's going to be a specific signal. Then it will interact with some other species in the reaction network, which are the program itself, like the function that is being encoded, and eventually interact with an output signal, which is usually monitored by fluorescence, so that we can see over time what's happening in the system. So that's going to be the result of the computation. So I'm mostly working in simulation, as Olaf mentioned. So there are multiple versions, like multiple models for the system. What is interesting is that from a single graph, you can easily derive a model of the system through, well, those equations. I'm not going to go into the details, but the idea is that from one signal, you only look at the templates going towards it and from it. And we have a different type of interaction called inhibition, in which this signal prevents the activity of this template. So you also take that into account, and you can automatically generate all the equations describing the system, simulate that, and yeah. So that makes the whole process of going from an idea, like a basic graph, to a simulation, well, automated. So a first result, well, some first story I want to talk about is one of the first attempt I made at evolving those controllers, so those chemical reaction networks. So I designed an algorithm based on, well, extended the NEAT algorithm that was designed for evolving neural networks to work with the DNA Toolbox systems. So the updated algorithm was called BioNeat. And the idea was that, we would set a fitness function and look at the system evolves. So in this work, which was presented at ECOL in 2013, I simply focused on having, two agents. So the agents remained abstract. I had no idea of how they would be eventually implemented, but just that they could communicate somehow. And they would play rock, paper, scissors. So a simple game. To play that game, I defined 3 specific species, so rock, paper, and scissors, that would represent the hand they're playing. And because they need to know what is happening, I gave them a reference to the other agents, rock, paper, and scissors. So the species of both are different, but since we're working with DNA in terms of implementation, it's like there is an ID part and a common part that just represents what kind of hand it is. So they can interact with the species of you're playing now. Like we look at the hand now. And the idea is that every time the clock ticks, we look at the highest concentration among rock, paper, and scissors for each agent, consider its hand, and give 1 point to the winner. And at the end, we make them play 10 times in a row. And at the end, it gives a score for that match. And since we're using an evolutionary algorithm, we have a population of agents. We make them all play against each other, take the total wins you have, and that's your fitness. And we keep the evolution going like that. What we were expecting at the time is that the agent would learn to play rock, paper, scissors, probably try to evolve some kind of memory, try to guess what the other agent was going to do. In the end, what happened is that they learned how to cheat at rock, paper, scissors. So it's actually exactly the same as I saw that an interesting video of like an actual mechanical robot using an ultra fast camera to see the slight movement of the hand of the other player to always play the winning hand. And it's, long story short, that's about how they are doing it here. So here's an example. So we have 2 agents. So to make it simpler, there is the bottom agent, which is here, and the top agent is shown here. The clock is in red, and those are, the color of the different hands. So for instance, this agent on the left, in this case, is always playing scissors. And this agent on the right, on the other hand, is doing something, kind of smart. So this one is playing scissors because it is actually monitoring the concentration of the other agent, right? It says, oh, if there's paper, I'm going to make scissors. If there's rock, I'm going to make paper. And if there is scissors, I'm going to play rock. However, if there's nothing of interest happening, I'm just going to default to rock. And this agent here says, you know what, I'm playing rock, right? Like, as you can see, the rock, so the green line is slightly above the paper. Wait, the paper line is. Oh wait, is it the opposite? So what happens is that okay, no, I understand. Yeah, I think the two are inverted because this one is playing, it ended up being paper, and this is scissors. Sorry, there might. Sorry, there might be I might be confusing something here. Rock and paper. Anyway, whenever there is a tick, the hands switch so that the other one does not have time to react. Yeah. Okay. Yes. Okay. Yeah. So this one plays. Sorry, this one plays rock. This one plays. Sorry, the green is above. So this one plays rock every time there is a tick. Like, it switched his hand at the very last minute. But this one, because it was just at the limit of playing paper, as there's a tick and detects rock, tries to, well, actually switch its hand. And this one tries to catch up, but fails because it is too late. So thanks to that, this one, the one on the right, overcheats the one on the left. Yeah, so they're both trying to catch what the other is doing, but eventually, you know, because the time frame is limited, only one end up winning. Here is a tougher example. So here I didn't split the two networks. I'm just putting them together and indicating one side with a dashed area. But what happened, as you can see, is that they're really trying very hard to outsmart the other agent, and to the point that they're even ignoring the clock. They're only fighting against each other, and you know, it happens that one side wins more often than the other one, so it is actually smarter than the other one. And yeah, so this is getting way beyond what was expected to happen when we first thought about this design. And it gave rise to interesting observations that, you know, the bigger you are, the more modules you can evolve, which allows you to cheat in smarter ways. But those modules means that you are going to use more the enzymes, so you're slower. So you have lower, slower reactions, right? So a less smart but faster agent can still beat you. All right, and so sorry for the poor quality. So if we look at the arms race of, you know, the base agent over time, so at first it's simply you look at one species from the other guy and you switch your hand based on that. Eventually you look at all the hands of the other person and switch based on that, and actually start having comparison in terms of concentration. So you try to compare which hand is at the top. Then eventually you start using the clock so that you can switch your hand at the last minute, and because you realize that the other agent is actually monitoring your hand as well. So you want to hide which hand you're playing, and at the last minute switch to confuse the opponent. And then eventually, so I don't have time to describe what's happening in this graph, but what's going on here is that you try to see a change in concentration of the opponent. So if the opponent looks like he's playing one hand, like let's say rock, you want to play papers, right? But if suddenly there is a rise in the scissors concentration, you may think, wait, that agent is going to change its hand at the last minute. I should actually play rock as well. And that's actually the last step we could observe. It's not clear whether we would have further steps going on, but yeah, at least there are that many layers of strategies that could be observed in the agent evolved. And the question we had then is, well, are they really getting smarter, or is it just some kind of blotting of the networks, and actually they're not doing anything in particular? So what we did is that over 10 runs, we took the best, so the agent that won the most at each generation, and made them play. So at this pool of 2,000 agents, and made them play against each other. And on average, we can see that the fitness, so the number of wins, increases until it kind of plateaus toward the end. So there is some learning happening, but at the end, the learning slows down. And that makes sense considering that first, it's looking at the agent, it's getting harder to find new modules. It's harder to be smarter. And at the same time, as I mentioned, being smarter means you're slower. So even a basic agent might end up winning. Like you're expecting so much out of the other agent, when it ends up doing nothing, you're actually surprised. Right. So just to conclude on this, what we learned at the time is that it's easy to cheat. So it was not in the paper, but if you disallow cheating, like you remove the reference and instead only tells the agent whether it won or not, they can actually evolve a memory to try to remember what was the previous hand and play according to that. But this is much harder to evolve, so it quickly hits an optimum. And by looking at what the agent were doing, we learned a lot about what you can do with the pen toolbox. In particular, we found like a few design patterns that we're still using to this day in terms of concentration comparison, looking at differentials in concentrations, and so on. Okay, so I'm going to stop here on the first part and move on to a different piece of work that I did at LC a few years ago, where we went further and had an actual implementation, like a basic implementation of molecular robots. So those ones were not moving yet, but we could experimentally have them produce signal and talk to each other. So we have and that work was based on a previous work from some of my collaborators, so Guillaume Ginès and collaborators. And we thought to extend that work. So their work actually had those beads with, as I mentioned, some just grafted templates from the pen toolbox. And they had two types of beads, some with the A to B template and some with the B to A template. So because of the exonuclease, if you only have one type or the other type locally, nothing will happen, right? Because even if the reaction starts, well, the exonuclease will degrade all the signal, and you will have only the template left. But if you have both A to B and B to A near each other, they can actually have an autocatalytic process that will produce both A and B. And we can observe that through fluorescence. So that's the output of the program, as I mentioned in the introduction of this talk. And what we were interested in was to think of using saturation, so abusing the enzymatic process, so that parts where there were only a few beads of one type could still start activating, even even though locally they were not that much templates. So in their previous work, if they have like a lot of template and everything, you can really activate everything. So the signal kind of propagates through the system like wildfire. But in our case, we were more interested in having only a few beads of one type, and using saturation to force the other clusters to light up. In simulation, that looked great, right? As long as you had enough polymerase, or as long as you produced enough signal to overcome the exonuclease. So while the exonuclease is busy somewhere, trying to kill signals there, it actually do not have time to kill signal somewhere else. So that other place, even with lower concentration of template, can start lighting up. But that did not do exactly what we thought once we did the experiment. So I'm going to show a video, and I hope everyone can see it fine on the stream. So you have mostly A to B beads, so all of those are A to B, and there's only one B to A here. So at first, well, it works as expected. The signal lights up, and well, in this case, there are literally no other B to A around, so there's no other cluster that can start up. But what we saw instead are those things. Looking more into it, those are actually parasites. So those are, like wrongly produced DNA strands. So DNA strands produced by accident by the polymerase that eventually reach a point where they can autocatalyze and form this kind of structure that are so big they're actually visible. This is about 100 micrometer of DNA produced out of nowhere. There are smaller ones as well. And those just keep growing until they have consumed all the fuel that is locally available. So the DNTPs that are locally available. So that was not the expected result, but it was quite interesting to see. So in the previous work, this kind of behavior was not present because they used much lower, probably because they used much lower concentration of polymerase. And so that even if something like a parasite was produced, there are still enough exonuclease to go around to kill those parasites before they could do anything bad. But that was not the case here. So in using that approach, we could have an emergence of some kind of strange molecular species. And that's fine and all, but we didn't look too much into it because parasites are very dangerous when you have an experimental lab, in the sense that if you mishandle it and it goes into, spreads around, basically all your experiments are going to fail for a while until you get rid of it. That's something, not this time around, but that's something that happened in that lab previously. So when we saw that parasite, we were like, oh, okay, we're going to throw this away and forget about it. This, makes sense in the sense that it was not what we are looking for at the time. Thinking back about it, it was actually a cool result. So yes, that's why I was inspired by the topics of cross labs and crossroads to talk about it today. But yeah, so I think I'm out of time anyway, so I'm going to stop here. I think the general conclusion is that we can use molecular programming for designing and controlling agents at the molecular scale. So mostly robot in my case. In general, which is the thing that I did not talk about today, we focus on rational design, and we try to keep things working exactly the way we want. But it's also interesting sometimes to let things, do their own thing and explore the capabilities of the medium. So, there's the famous sentence, it's not a bug, it's a feature. So that's how, those cheating agents give us some insight on what can be done with the DNA Toolbox. And also, sometimes we have unexpected behaviors such as the appearance of those parasites that could make structure that's so big they're visible at that scale. And that could be used as a breadboard system for open-ended evolution, maybe. But anyway, so that's all I have for now. So yes, so I'll be happy to take your questions now. So if there is any question.
Thank you, Nat. Very nice, and very fun work also. Playing. This makes me think that there is on Kaggle right now, for those who are Kagglers, a little competition on rock, paper, scissors without cheating, but with memory. So you can submit your own algorithm if you're into that. Of course, the dynamics here are interesting because you're allowed to play on the cheating part. So there is there is a link I posted also, and I think that's what you were referring to. I'll post it here too if I can find it, about the cheating robot. I think it's a Japanese lab, right? Is it that one?
Yeah.
Yeah. Let me see if I can find it. I pasted it somewhere. Oh, I think, yeah, I got it. Oh, lost it. Got it again. Yeah, there we go.
So it really looks like a kind of wireframe hand, and it does this, and it's always the winning hand.
Right. Yeah. So yeah, which is very frustrating if you are a human. But I've never seen the version that plays against another robot. So maybe they can take advantage
actually,
of something else.
Yeah, because of my work, the first time I saw that video, I was like, oh, what happens if you put maybe they're expensive, I don't know, but what happens if you put two of them in front of each other?
Yeah.
Right.
That's right.
I assume there was some small differences in the material, so they would not always be able to win. I don't know. Do they always draw? Okay.
Right. I actually, for those who are watching this, what's and maybe they're not familiar with DNA computing, what's the time scale at which you are playing those games? Can you watch?
Right. So let me go back. So this is the time scale is here. So minutes. In minutes. So you have this is about I think 50 minutes. One hand is 50 minutes. So it's slow. It's not, you have to be quite patient if you want to play manually against the systems. You can actually. So you can put this in a test tube, and it will actually, well, I guess if you put only one side in the test tube, it will do not much. It will just do its own thing. And just when you inject the clock, you can inject your own hand, for instance, like what you want to play, and it will still react as usual with the system. So you could technically play against it as a human, but yeah, you have to be quite patient.
Right. But this is really a great model because it shows in a very simple system of reactions how you can have already a mix of competitions that create this kind of triangle arms race, right?
Right.
So maybe that's. Did you make an argument ever about this and well, origins of life kind of chemistry?
No, actually, I don't know. No, I didn't think about that specifically because, yeah, this work was, you know, I was not in the mindset of looking at the origins of life specifically when I was doing this, when I was working on this. So it was more in terms of, what can be done with with molecular programming, and in particular, what can be done with the pen toolbox. So at the time this was published, the pen toolbox was really at its beginning, right? Like we had two systems that were implemented in the pen toolbox. We had one oscillator, and we had one memory, right? Like we knew it was possible to have memory. We knew it was possible to have like dynamics, but that's about it. We didn't have that many insight on what could be done, and especially what could easily be done with the pen toolbox. So yeah, that was really interesting to first, look at the time series of those systems and be like, what's going on? And then look at the network and be like, oh, I still don't understand. And then really look at each part bit by bit and compare them with each other and be like, oh, okay, this comes back often. This motifs come back often. And okay, yeah, it does this. All right, like I don't know, like okay, so this will, because you have those comparison in a row, or this one is actually interesting. Like you have two autocatalysis, but one is optional. Like if something triggers this inhibition, you would have actually a second level. So like if there's no inhibition, you have a high level. If there's inhibition, you have a middle level. And if there's too much saturation so that this one cannot start, you have actually a low or almost zero level. So instead of being 0 or 1, like, binary, like we would usually have so far, suddenly we had like, 0, 0.5, and 1, right? So, and in hindsight, it's kind of trivial, but at the time we didn't know what we would be able to do with the pen toolbox. So that was really what I was focusing on.
Yeah, no, it makes sense. So you have the kind of flip-flops kind of elements that you get in normal computing. So maybe before we answer questions, can you give maybe what is it, like not a primer, but I guess explain what is the difference between regular computing, what I have Zoom running on right now, Firefox, Discord, whatever applications, and can we run that on DNA computing the same way? And what is the advantage of running things? What is, so I was always excited when I learned about DNA computing in a computational complexity class. Oh yeah, DNA computing is actually amazing for certain things. What are those things, and what's the difference?
So it depends on who you are asking. So there are some programming languages of DNA computing. So for instance, DSD, so DNA strand displacement cascades, that are known to be Turing complete. There is no proof for the pen toolbox, but we suspect, so there is no peer-reviewed proof for the pen toolbox, but we also think it is Turing complete. So anyway, any program you can, in theory, implement it with molecular programming. However, it's not appropriate to general computing, I think. The main reason is that, so molecular programming is like a massively parallel system. You have all those molecules, so you don't have local variables, right? Like all the variables are together and they always want to interact together. So basically you always have operations going on in parallel at the same time. You don't have any critical area. You don't have, it's like a parallel computation nightmare. So for that reason, it makes it extremely tedious to make like a complex, like to make a program the same way you would code in C or something. And then, so I see the question of Julia in the middle. I'll get to that later, but there's also the reality gap, right? So even if in simulation you get the correct behavior, once you try to do it in practice, you need a lot of tuning before getting to the correct result. So there are some systems that were shown to work correctly with molecular programming. Like there are some linear classifier. There are like stuff like perceptron, things like this. There you have one that computes the square root function, but only up to 9, I think, or 16. I don't remember. Because just scaling up, scaling further would increase the number of molecules, and it's beyond what can be reasonably expected from a human to do in vitro. So
what would be the most impressive result that for you personally? Done DNA computing.
So that's why I'm mostly focusing on swarms, right? Because in swarms, you're fine having a very simple agent. So having a simple program is fine, but you use the number and interactions between those agents to actually have a complex behavior. So for me, that's the appropriate, I don't know if it's the best thing you can do with, or the most impressive thing you can do with molecular computing, but for me, that's the one that is the most targeted toward molecular computing. Because if you're trying to use a kilobot, like a kilobot is, even though it's not that expensive, it's still quite expensive, right? Like you cannot have 1 million kilobots.
Yeah.
But we can easily have, like we have hundreds of millions of the robots I was showing. So if we can program them to do something interesting, then we have the scale, right? So the scale is on our side. What is not on our side is the complexity of the program we can have. So yeah. So for me,
Plus, I guess
massive
that's right. So I guess there are many consequences outside in terms of applications. I think one like smart materials.
Right. So smart materials in particular. So we're working at the molecular level. So one thing that can be done is really like sensing molecules that are present in the environment, like whether there are some toxins around, and you could interact with that and change your behavior so that there would be aggregation there. So sorry. So in the example I had, you would have, taking the example I had, you would light up where there is something dangerous. So you could spray your robots on the surface and see, oh, there is corona here. Coronavirus is here. Okay, I should really clean that up.
In theory, there are many possible applications. But I think so. Some people are building them, right? Like those actual molecular robots. Maybe we can talk about that later. But let's take some questions, maybe.
Right.
How about the first one?
Competition between agents. Do you have example of collaboration? So the first question. Actually, the second example is an example. It was supposed to be an example of collaboration. I did not show the correct case. I did not show the case where it works. So I didn't show when there is actually collaboration. But the idea was that you have, when you have both types of agents, they try to light up. But because there is a lot of exonuclease, they usually cannot accept if by chance you have a lot of those agents locally. And with the correct setup, if you don't have, well, you would aim for needing at least 2 or 3 of both types together to start the reaction. In the case I was showing, just one agent was enough to light up. But yeah, so you would have places, so cluster with large number of both types of agents, and they would light up. And then further away, you would have a cluster with less agent, and normally they would not light up. But because this one way over there is lighting up, like suddenly, their exonuclease has a ton of work to do, so it cannot stay here. Well, it cannot focus too much on the other cluster, and because locally the active concentration of exonuclease decreases, suddenly we pass the threshold that allows activation. So it actually can light up. So both clusters are actually collaborating, and because and actually the idea was that once both light up, you would allow even further smaller, smaller cluster to light up, and so on, until eventually hopefully everything that is close enough to another cluster would be lighting up. So yeah, it is supposed to be an example of collaboration, and just that there's this nice collaboration going on, and there's this parasite that just said, no, you know what, I'm going to do my own thing.
Right.
So
yeah,
then
and,
the reality gap is generally a problem for evolutionary robotics. Do you think about how a robot could transfer to reality? So yes, because we actually, so especially while I was at LC, we did some actual implementation of robots, not only the one I was showing, but actually further implementation. So with actual robots that actually move, so that are smaller and move. So that one was published in the Gecko 2017, Gecko in 2017. And yes, it is really hard. In practice, so I was showing, like those networks with like tens of templates. In practice, we found that beyond, so it's been a while already, so I don't remember the exact number, but I think beyond 3 or 4 templates, it's really, the different saturation and cross interactions starts to really make it diverge from the expected behavior. So that's why in each subsequent work, we've been trying to put a constraint on the total number of template we can have. And yeah, that's one of the active direction of research we have right now, like trying to bridge that reality gap. We do need more experimental data now. Like at my current affiliation, I do not have access to an experimental lab directly, so it's a bit more difficult to try to see what's going on. But yeah, so we have some ideas about, so we know it exists. We know roughly, the safe range for systems. And we also know that for most of the application we have in mind, we need more than that safe range. So yeah, we are considering different ways to bridge that reality gap.
all right. And another question about parasites.
Always appearing this experiment, special conditions need to be met. So yes, so I guess both are a yes. So you need special conditions, but if you have those conditions, then it always appears. But in practice, if you are working in 0D, in a well-mixed system with some autocatalytic templates, I think if you wait long enough, eventually it will always, I'm not 100% sure, but with a very high probability it will appear. But yeah, in practice, yeah, you need some amount of polymerase and some autocatalysis going on in there. And specifically in those systems, if we had lower concentration of polymerase, it did not appear. But as soon as we had, so yeah, I don't have the numbers at hand, but as soon as we had like a specific concentration of polymerase compared to the rest of the system or more, we would see them appearing. Yeah. So yeah, definitely there's something related to how much polymerase you have and how much exonuclease you have. And probably the type of polymerase also matters because I think the one we were using is faster. So we want, to produce quick, the reactivity to be high. So we want a fast polymerase, but because of that, it might make more errors. So I don't remember exactly which one. So we were working with multiple types of polymerase. So yeah, it might also be due to the amount of errors that polymerase makes.
Yeah, it's a very interesting effect. I think we should try and replicate it. See if we can get it to happen again in various conditions.
So I remember. So I was at LC at the time, as I said, and I remember talking about it with some other experimentalists working on DNA computing there, and they said, "Yeah, sure, you can replicate it, but somewhere else. We don't want that here. Don't bring a parasite here."
Yeah, parasites are considered again. It's not about lack of interest; it's really about the danger that they would infect the whole machinery.
Yeah, especially there was, as I said, we already had experience with parasites when, for some, because of mishandling of some experiment where a parasite appeared for, I think, 1 month. So even though we tried really hard to clean everything for like 1 month or so, we could still, basically nothing was working, because even if you put nothing in the tube and just the enzymes, like you would have like autocatalysis because the parasite is there. And we did not know what was contaminated, but something was contaminated. Probably the buffer, I don't know. And so you had to throw everything away, clean everything, and start again. So they were like, no, not again.
Yeah, this is again a nice demonstration of the the patterns of serendipity and randomness in science, where most of us try to push away certain types of errors that happen, but you studied one of those errors, and it became interesting. And I expect this to be omnipresent in science in general, where you're just not looking through the right lens, and you see those parasites as and I understand that they are very nocif for
the usual application, yes,
for your experiments. But in this reframe kind of interest, they became really interesting, and you would like to know more of and how they behave, maybe how to control them better.
Yeah, in practice, in particular. It would be interesting to see the sequence, right? Like to sequence those parasites, like what are they made of? Like why do you make this structure? Like how do you polymerase that way? And especially if you're polymerizing, that means you're technically in a configuration that would prevent polymerization, but it still polymerizes. So what's going on in there? It's kind of
right.
But yeah, at the same time, you don't really want to mess up your world lab. Like probably you would need to have like a specific, like very clean setup, experimental setup, where you're separate from everything else, and you have like equipment just for that. So it might be.
I guess. Then you might be missing it, right? So certain effects in physics happen just because because your utensils are imperfect, and if they're not.
That might also be the case. Yeah, maybe there is actually all of this only started because there is some cross contamination from some other experiment that was done before. Maybe you don't know actually.
Yeah, you don't know. Right? Maybe you need some kind of it's more than symmetry breaking here. It's really the existence of some kind of species, even in small amounts. I had a question about theory of mind, to bring this back to intelligence, I guess. So I guess your agents here in the first experiment, the rock paper scissors, they are trying to guess in advance, right? Like what would they do? What would the opponent be moving next?
Yeah.
And they try to incorporate. Yeah, so what would it be to incorporate actually one level down this sort of inception path? You know, like going one more degree into more theory of mind. I think that you think that I think, etc.
Right.
What's the limit in terms of the chemical system? Do you think we can go very far?
Well, so far, what the best agent we found could do was to first look at what you're doing right now. So what you're thinking right now. Try to check if you don't have other concentrations that are close enough. So try to check if you're not thinking about switching your hand later. And also check whether you are moving your hand. So if it looks like you're thinking about doing something else, like whether you're trying to switch your hand just before the limit, then it would also react. So going one step further would be to beat that strategy, right? So it would be to look like you're thinking of changing your hand at the last minute, but not switch it. Right? So like you would trigger the other guy, so he would switch his hand, but because it's so big, so one thing is that the bigger you are, as I said, the slower you are. So if you commit to one hand, going to another hand, it gets really hard. So you would actually force the other guy to commit to the wrong hand just before the clock, and then actually stay where you are and win. And going one step further than that would be to delay committing until the very last minute and really try to look at the gap between the concentration or something like this. I'm not quite sure.
Well, I guess at some point it's difficult to differentiate between a theory of mind in this case and a cultural effect where everyone is doing this switch at the very last minute possible.
Not everyone, right? So because especially that we were thinking whether or not there was this kind of cultural effect. But if if you make the agent play against other agents that were not doing that, they still win, right? Like
they do.
So if you play against something who's dumb and just play rocks all the time, well, you're going to win, right? Like even if you think that it's weird, is not switching its hand, but you would be like, well, okay, I win.
I see. Yeah, I see. But if they were all using this effect, then it would become cultural, and you could play on that because everyone else is doing that thing.
Right.
You can take advantage of that. But before that happens, you're right. So if not everyone is doing that, they can take advantage of it.
Yeah. So, but actually, the fact that you're looking at whether the other person is switching or not already comes from the fact that that people are switching their hands at the last minute.
Right. Exactly.
So but even though, I'm still going to like I'm anthropomorphizing those agents, but there's but anyway, they're at the end of the day they're just networks randomly it's randomly updated until something happens. So it's hard to say that they're actually thinking. It's just they're not thinking, right? It's just a simulation. That is based on some equations. But so we can interpret it as being this kind of strategies.
Right. And if so, a different question is even with those effects, and they are cheating, but at some point they're all cheating to the best extent they can.
Yeah, very quickly. So basically, while you're not cheating, you have this standard approach of everyone plays if you have a majority of agents playing rock, then you will have a majority of agents starting to play paper. Then you will have a majority of agents playing scissors, and so on, right? Like it will just go through loops. Then suddenly you discover, oh wait, I can actually cheat, and you would start increasing.
But in that case, so if they do that, you said that you could they could evolve a memory or those kind of hysteresis effect. Do they evolve them if you don't disallow cheating?
So they do evolve a memory if you don't allow cheating. If you don't allow cheating, or if you allow cheating, they do evolve the memory. But a memory is much more, so there's like an evolutionary cost to getting that memory, because a memory takes 4 templates. So you need at least 4 mutations to get to a memory, while cheating can be done in 1 template. So while you are evolving the memory, like 1 bit of memory, the other guy can evolve, like it can actually monitor all your moves and has still one template left to do something else.
Oh, so I see. Not an evolutionary cost, but really a computational, like a time and chemistry.
Yeah. So when we look at the total set of agents, we could see that memories happen. Like there are some agents with memories, and they're doing better than like a completely random agent. But they are completely outsmarted by cheaters. Even cheaters that are smaller than them, and smaller also means, as I said, that you're faster. So, they're just completely outsmarted. Well, outrun.
It seems very important that you are able to design systems, chemical systems, those molecular robots too, right? Such that they can form soft memories very easily. And then correct them within the time they are solving tasks. So it seems like a very important problem to solve.
Yeah. So in that case, it came from the design of the experiment. Yeah. Again, if you realize that the problem comes from cheating and you prevent it, they will actually go toward soft memories. The interesting thing about the pen toolbox is that indeed it's actually quite easy to design such kind of memories. So that was one of the reasons why at the time I chose to work with the pen toolbox rather than some of the other programming approaches.
Yeah. I see. Yeah, that's that seems very important. Maybe one last question. I think this one. So if parasites appear so easily in the lab, can we really develop practical applications in the real world? Based on molecular robotics reliably.
So yes, in the sense that they appear under those specific conditions. Well, those conditions that I mentioned, those are simple conditions, so you can easily get parasites, but there are ways to prevent it, right? So one thing we know about some of the parasites is I think they were 80 reach or something like this. I don't remember exactly, it's been a while. But there are some other enzymes you can keep in your system that will prevent the formation of parasites. You can play with you can keep your concentration of polymerase low, for instance. You have specific designs. So there are some people specifically researching how to prevent the appearance of parasites, and there are some sequence designs you can do to, like, you can play on the sequence design to at least delays the appearance of those parasites. So there was like a paper last year on that specifically that I saw. So it is possible, like, you know, the parasites are one of the things we try to keep away from our designs, and people work on it, and it's actually possible. Like, people in general can easily manage to deal with parasites. So so you can get to a point where your system will run out of fuel before a parasite appears. So in practice, that means that you would, you know, for a real life application, you would still need to replace the fuel at that point. So you will clean the buffer at the same time, which will remove any traces of parasites. So you would reset it. So anyway, for a real world application, if you design it correctly, you can avoid the parasite. And well, maybe it may still appear, in which case your system is broken. But you know, if you drop your iPhone, it's broken as well. So it's a thing that happens.
I guess, yeah, we have to find ways for both systems to be robust, I guess. All right, maybe let's move on to the more casual discussion and go offline for that. Again, thank you so much for joining us from Ochanomizu University, right? And let's keep on discussing. We'll share the link to your lab. So you don't only do this work, but this work actually is from a bit before, right? You also do other work linked to, I guess, evolutionary computation, robotics,
right.
I mean, molecular robotics and DNA computing.
Yeah.
Thank you so much then.
Okay.
All right. So I'll find the way to end the stream, and we so before that I'll share the link to join us on Zoom for those who want to. If I can find the right one, this one. All right, and see you over on Zoom. And the stream.